My name is Mohammed Bawena. I'm the Associate Area Head for Biological Sciences. I welcome you all attending the Bill Brown Distinguished Lecture in Biological Sciences. And to start this lecture, I would like to invite and introduce the Dean of Carnegie Mellon University in Qatar, Dr. Mike Trick.
Thank you. Assalamu alaikum. Good afternoon and welcome to Carnegie Mellon University in Qatar. I am Michael Trick. I'm the Dean here at CMUQ, and I'm pleased to introduce today our guest, Dr. Carrie Doonan from the Department of Biological Sciences at Carnegie Mellon's campus in Pittsburgh. Dr. Doonan is teaching professor at the Mellon College of Science and serves as Director of Undergraduate Laboratories, which understates the impact she has had on laboratories at Carnegie Mellon. Before I tell you a little bit about Dr. Doonan's expertise, I'd like to note that today's presentation is part of our Distinguished Lecture series. Distinguished Lectures at Carnegie Mellon provide a glimpse into the depth and breadth of scholarship of many experts who visit our campus. We have different types of distinguished lectures, each representing a field of expertise, and each type is named after a notable academic from Carnegie Mellon. The Distinguished Lecture in Biological Sciences is named in honor of Bill Brown, longstanding member of the faculty at Carnegie Mellon. Dr. Brown spent more than 3 decades as a professor at the Mellon College of Science, serving as head of the Department of Biological Sciences from 1995 to 2000. He was a visiting professor of biological sciences at CMUQ and was named a special assistant to the first dean of CMUQ, Chuck Thorpe. Sadly, Dr. Brown passed away in 2007, but over his many decades of service, he made an indelible mark on the way biological sciences is taught both at the Pittsburgh and Qatar campuses. It's fitting today that today's Bill Brown lecture is being delivered by Dr. Doonan, who has dedicated her career to fostering excellence in teaching in biological science at CMU. Dr. Doonan's primary area of focus involves the teaching and administration of experimental laboratories. She writes experimental units, trains junior faculty and teaching assistants, and is actively involved in all aspects of undergraduate program. Dr. Doonan has adapted many of her curricular innovations for use in K-12 outreach and has been invited to present this work at many regional and national forums. She has served as a Biotechnology Institute National Biotechnology Teacher Leader, received many awards for her innovations in teaching. Please join me in welcoming Dr. Carrie Doonan to the Distinguished Lecture Series at CMUQ.
Am I on?
Yeah, it's not.
Okay, I'm great. Thank you so much, Dean Trick. And thank you for allowing me to come and share a little bit about my experiences here at CMU. And let me just say that this is my first time here and how much I really, really enjoy being here, meeting everyone, talking with everyone, and learning all about this campus and what a unique and special place it is. There are some similarities. CMUQ, the students are just like CMUP. The instructors, the faculty, the staff, everyone wants the best education possible for our students. We want to educate the next leaders, the future leaders in biotechnology and medicine and in industry. So as I was preparing for this talk, I was wondering, what experience do I have? I've been here for 31 years. Actually, Bill Brown hired me and I thought, what could I say? How could I organize my talk so it would be beneficial to both students as well as faculty? So who do I turn to? I always turn to the students and both alumni and I said, what should I say? Please tell me some tips. What should I say? And the inspiration came from a recent graduate who was a CA for me. She is now second year Duke graduate school in the program. And she said, Dr. Doonan, this is what you need to tell everybody. She says, I'm in my second year Duke PhD program in biological chemistry. She said, every single time in lab, and I complained about it bitterly, you made me write and analyze my data and say, did my data meet the expectation or not? And I had to explain why. Every single time I had to design an experiment, she said, I use that today. And it's why I'm in charge of 5 other TAs. She said, let everybody know that and share how much problem-solving ability that we have. And that's where my inspiration came from. And so I'd like to begin by, a little bit about me. I've been teaching labs for 31 years. I love lab. I'm there all the time. I really like it because lab is a place where anything can happen. I often joke, I can put out a fire, I can talk on the phone and do anything, but I absolutely love being in the lab. I've been here for 31 years. I teach genetics lab, I teach the biochemistry lab, I've taught the FADS lab. I even, for fun, one summer decided I was going to teach the bioelectric class. Wow, what a challenge that was. But like anything else in life, you learn from that, and then you take that with you, and you take that data with you. Anybody else who knows anything about me knows that, please, when you come to Pittsburgh, visit my office, because the food is always there, and I am famous for my brownies, okay? And I will happily give anybody brownies. Just let me know that you are coming. In fact, in lab class in the fall and the spring, we have a brownie contest for the highest and most pure concentrated DNA. Lots of fun. Like to introduce things in lab, but I didn't always start that way. Bill Brown hired me, so I thought, problem solving. Let's talk about Bill. Bill was the head of the department from 1995 to 2000. He was a talented biochemist who studied proteins. He did a lot of work cloning Taq polymerase and also worked in occupational asthma. He was a visiting professor here. He was so passionate. Bill even communicated to me that this was the place, it was special. He was coming here because he loved it so much, and he thought that establishing the labs and this problem solving and training our future scientists was so important, and this was a special, unique place for that to happen. It was very unfortunate when he passed. In fact, the whole department was devastated. This man who was passionate and a leader and talked to everybody and was a friend to all was really, really a hard time. But one thing about Bill, and that is we need to look back at Bill's vision for the biolabs. And we know that Andrew Carnegie, he founded Carnegie Mellon. And I found a quote that says, it is a university of problem solvers, back in 1900. And I think that's the common theme here. So Bob Rice on the CMU faculty led the effort to create a new bio department focused on molecular bio, cell biogenetics, biochemistry, and biophysics. This new department flourished and made major contributions in the basic sciences and biotechnology, and even then looking at developing new tools and computers. In fact, I found a quote from a book that's called The Innovative University, published in 2004, that says that Carnegie Mellon is a jewel among the world's university. It radiates powerful teaching, interdisciplinary research, public service, and a persistent curiosity about things. This is what makes this place special. We foster that curiosity. We want that. We want those problem solvers. We think about that. And why I've lasted 31 years, I never thought I would make the first semester when Bill hired me. And I'll get to that in a minute. But I love teaching in a place, in a lab, that has all the equipment there, that we have funding, so our students know, and that's why I play the Price is Right in lab, not to scare our students, but to let our students know that they are using all the lab equipment that they wear. We are training them to go out into the world and be a research technician or be the next future graduate student. So let's get back to Bill. Bill Brown started the lab. So students, sorry, that's who you have to blame for the labs was Bill. Bill said, and it was always his vision, that the labs were to be hands-on lab experiences, not cookbook labs, but they could not be predicted outcome. They were to be hands-on, creative lab experiences that would help promote and gain student interest in research. That was Bill's vision then. And that was probably, I couldn't find the exact timeline, but in the late 70s, because there's been one other in Pittsburgh, director of undergraduate labs, that was Linda Kaufman. I was hired because our biology department grew so big that we needed another instructor, okay? Bill was also a big component in fostering a bio community, a community of learners, and he helped, and this is a quote from Amy Burkert, our Vice Provost for Educational Initiatives, who worked for Bill, and he helped her get the BioSAC, that's our Biology Student Advisory Council, just like you have here, building efforts that went underway that set a tone, community of problem solvers and learners working together, and that. So Bill's vision, imagine my first semester, there was already a director of undergraduate labs, was there doing labs my first semester. I was like, oh my goodness, am I gonna make it? I didn't know if I was going to make it through December. In fact, I thought at that time, when Bill hired me, there was a clause in my contract that if I made it through the end of the semester, I might get another contract or something, but that's to be debatable. But anyway, Bill's vision. His vision was active learning. Bill's vision back then was what we know today, what we know about education and that active learning. So I always like to go back and look at the definition. It asks students to engage in their learning by thinking, discussing, investigating, and creating. Students who are more engaged in the material are more likely to retain that material and carry it with them. And this applies to lab and or lecture classes. In lecture, a broad range of teaching strategies which engage students as active participants in their learning with the instructors. You can look online. You can find a lot of studies that says, does active learning really make a difference? I always like to look at the data. Anytime I'm faced with a problem, I look at the data and see what I can't take from that. But two such studies spoke to me. One is a PNAS study that says active learning. Students who are learning in an active learning class in exams, their exam score increased about 6% in active learning sessions, and students in traditional learning classes were 1.5 times more likely to fail. Amazing to me. There's a CMU study from CMU's Nezra Yanier and Ken Codinger from psychology, and AI that was published in Science that says AI tools and that, but active learning really makes a difference. So knowing this and realizing that Bill's vision for this, how does this apply to how we do active learning in the labs today? And what can we gain from that? And how can we learn and adapt to that in lecture classes? We do know, it's stated everywhere, that active learning improves retention, increases your critical thinking, collaboration, increasing engagement. And I've seen this a lot. Students tell me all the time that the lab courses at CMU typify their hallmark of CMU education. And they improve analytical, critical, and problem-solving skills. Secret, one of the reasons that always inspires me to continue doing my job is the nice folder on my computer that says, of all the students send me messages and say, Dr. Doonan, I never thought I would be writing to you, but don't change a thing. Today, I taught the postdoc in our lab what to do. So that's what inspires me. That's what inspires me when Bill had this vision for active learning and the lab classes. So what I'd like to do now is just take a minute and go over what are some types of active learning, and have you seen these before in your lecture classes or your lab classes, and how do we focus on them in lab? They are think, pair, share, jigsaw discussions, One sentence summary, muddiest point, minute paper, sharing activities, problem-based learning, and simply pausing for clarification during a lecture. I've tried some of these, some worked, some did not. But lab, can we map these skills onto what we do in lab? Group work, designing and proposing experiments, data analysis, troubleshooting, scientific writing skills, sharing activities, hands-on problem-solving skills, providing a supportive learning environment with the goal of increasing engagement. Lab is active learning. So let's take a couple of these. One, what is the jigsaw discussion? The jigsaw discussion simply is, if you think of the pieces of a jigsaw puzzle, they all have to fit together equally, right, to accomplish a task. So in the lecture class or in the lab class, when we put students together to solve a problem, propose a hypothesis, solve something, every single piece of that puzzle must fit together. Everybody must contribute to that learning, to that common goal. How do we do that in a lab class? We design experiments and we carry out multiple steps in a group. Now, I would love to, if I had time, I'm going to contact Everly about quantitating this, but what type of outcome does this have? The best thing I could do is, when I asked the students, what should I say? A lot of them said, Dr. Doonan, send quotes. And so I have a couple here. One is from Renee, who is working at Woods Hole right now. And she said, the lab experiments that I struggled with required me to think critically and outside the box are now the reason why I can piece together solutions and that. And Miwa notes that CMU pushed me to be a critical and methodical thinker. The next thing I'd like to talk about is data analysis and think, pair, share. Maybe you've done it. I do it in lecture class. I personally like this one. Think, pair, share is presenting your class with a problem, giving them a few minutes to answer questions about it, and then sharing it in the class. Sometimes it's a very effective teaching tool, sometimes not. How do we map this onto lab classes? We have to critically analyze the data from an experiment, match it to the predicted outcome, and we also do data analysis during the lab class, and sometimes that allows us to problem solve and troubleshoot during the lab. Those are my most favorite days. And that too, sometimes I ask my staff, somebody came up with a great idea, can you mix me 100 mils of this solution right away? I'm so appreciative when the staff will do that and honor that. So what do the students have to say? Ashwarya, who just wrote me, got accepted to medical, says, Dr. Doonan, I used everything I learned in lab and data analysis, and I've identified a possible storage defect in fibroblasts. She's studying this disease state, and she's going to be published in a paper. Next, writing and critical thinking skills. And note how all of these, I can't tweeze them out separately, because all of these skills together overlap. And they overlap in lecture classes as well as lab classes. So a couple of these, one sentence summary, challenges students to answer a question, who, what, where, when, and why, in a minute. So can you condense your writing? Do you know what you accomplished in that 1 minute time frame? There's a skill to doing that and condensing and being able to cut to the chase, because no one wants to read a whole page when we can read just a sentence. Another technique often is called minute paper. In a minute, students are asked to consider what's the most important thing they learned today and which is the thing that's the least clear. Sometimes this is done as an exit ticket in a lecture class so the instructor can follow back up. Muddiest point. Actually, when I came here, I heard Adviti say that, and I'm like, oh my, what is the muddiest point? I have to know this, I'm behind. And that too. But muddiest point is students are asked to write down either the most confusing thing, so the instructor can also go back and revisit that. In lab classes, writing skills are improved. We are always asked, you're asked to keep an organized lab notebook, having a flow chart in your notebook, and coming to pre-lab with questions about the protocol really improves your critical thinking skills. And what do our students have to say about that? Maya English says, I complained about this so much. I didn't want to write those lab reports. I didn't want to write the purpose of every solution. But she says, I'm able to troubleshoot my protein purification. And most importantly, she goes, CMU is uniquely good at teaching this kind of writing, troubleshooting, and abstract thinking. It's the goal of every instructor that I come across. Every lab instructor, every lecturer, that's what we want to do. We want to improve this problem-solving skills. Next, writing. Eric, who's defending his PhD, said, that was the best quality I learned. It came in handy for that. And Julia says, learning how to work together and navigating those lab reports really made a difference for her. Okay, another thing, sharing activities. They really promote communication and teaching. We all know that as a CA, you improve your communication skills. In lab classes, in the spring semester of Biochem Lab, you do multiple presentations. Anytime a student has an opportunity to speak, present, communicate in a small group or a large group, that is going to make them a better communicator and help them in the future. Also, we do some silly things in lab, and what we do are, we give, I talked about presentations. In Pittsburgh, we have the advantage that the FADS lab class for first years, the first year research course, takes place the same time as our third year, the genetics or biochem lab, and we have what's called sharing days. And in those sharing days, the students come and they sit at the table, and they give some prompts. They're there for about 20 minutes to a half an hour. But those conversations are, tell us what the best class you've taken, what would you recommend? And then we ask them, hey, the first years have isolated plasmid DNA in molecular bio for what purpose? And the juniors will say, I've isolated plasmid DNA because I'm going to map the location of my mutation in yeast. And they really share, and being able to explain, those days are my favorite days. Just walking around, hearing all the sharing, and then all the side conversations. And then the big benefit is I'm walking down the hallway in lab, and I'll see those interactions, and they'll say that. Or the other day, Keir said to me, can you believe it, Dr. Doonan? I was up on campus, and my FAD students said hello to me, and they weren't embarrassed of that. It's working. Sharing, communication, community, and that. We also do something else. Some of you, I recognize, have done this in the fall. If you came in the fall in genetics, do you remember this? We both did a transformation, and we had an exam on that. So we set up little stations, and your FAD students and the genetics students went together and solved problems, and they got a treat when they go around. Any type of activity that promotes learning is a great thing. Next. Communication and teaching. Communication and teaching, it's so important. Think about it, students, about being a CA. The skills that you gain by being able to explain a concept helps your own understanding, but really helps you further down. And my current CA in Pittsburgh, who just wrote to me and said what was going on, said, collaboration in science is all about this. The explicit, technical, and implicit communication skills I've learned come to mind on how I have grown in my years at CMU. So anytime instructors have asked you to explain, give a short talk, work in a group, those experiences, those skills are invaluable. There's some other additional skills that aren't mapped onto those active learning. And that is, I wanted to put this quote in here by Dan, who says, CMU is unique. It pushes us academically while providing the support we need. And labs provided that ideal environment. And that's what all instructors strive for. We want to foster an educational environment that's conducive to learning by everyone. And that my motto is always, we must be friendly and cooperative, always. Always. And Ona says this best, when we have to be willing to collaborate with our peers, because knowledge means close to nothing if you can't work together and communicate. Time management. Remember, students, the first lab in the fall, when you were asked to do a viable count, how long did that take you? And we put you back in the lab now, how long does it take you now? By doing things and learning how to write concisely, it really improves your time management. All of these skills do and make a difference. Are there problems with this model? Sure. There's complaints. I'm the complaint department. Some days I think I have a note on my back that says complaints, but that's okay. I can take that. At the beginning, it takes a longer time because a skill to master it takes a longer time. But in the end, the benefits are well worth it. It also takes a lot of time and interest by faculty, whether you're in a lecture course or a lab course, to make sure everything's going according to plan. The first time I did think, pair, share in the lecture, my lecture ran over 20 minutes. I got a lot of complaints. I did not know how to time it efficiently, so it was useful for class. So it comes with time, but there are that. And finally, the other thing is, and I see a lot of this, and that's the imposter syndrome. I don't know if anyone has dealt with that, but it is students who are unable to believe their success is deserved. They don't feel like they've earned the place in the lab. And that's so important. Fostering a collaborative environment where everyone feels comfortable to make mistakes in that. It often results in a lot of anxiety. And we ask our CAs, our faculty, we're looking out for that. We really try to help our students and foster that positiveness and that confidence by looking for evidence, telling that student, what a great thing you did today. You designed that experiment. You did this. Sometimes just doing that can make the difference in someone's attitude, outlook, and willing to go off the edge again and make that decision, okay? Also, we try to match people up with mentors. I could talk about this for all day, but environment is so important. I've talked about providing a safe environment, but it's really important in the lab. It is not a test. It is not a test. In order to everybody makes mistakes. Every great inventor made mistakes. It's usually the third or fourth time that great scientific discovery was found. But fostering an environment where it's okay to make a decision, and this may be wrong, and there's no points off for that. But where the points come off are reasoning the reason why. What happened? That builds your analytical and critical thinking skills. And no matter what class I teach, that's what I'm always looking for. I'm looking to organize it that way. The final thing I'd like to take a minute and say that I think is so important in education is there are no labels allowed. Every single person in this room may be having an issue right now. And if I could use my own self, I was quite nervous giving this talk. I am a 2-time cancer survivor. I had parotid cancer where I had my 3rd and 4th couple of cranial nerves removed. I thought I could never teach again. My joy, my love, my passion. And then 5 years later, as a result of my treatments, I had thyroid cancer. Some days I feel sorry for myself. Some days I feel like I don't want to get out of bed. However, it was the positiveness of the people who told me that you can do it, we can understand you. The first cancer operation devastated me. I didn't think anyone would be able to understand me or anything, my passion. But it is, you just never know what anyone is going through. And that's why it's so important every single day to give everyone the benefit of the doubt and give them a positive word. It's so easy to focus on that negative word or so-and-so was down today, but it takes extra effort. And sometimes that's all people need. Again, environment makes such a big difference. The final thing is, in addition to environment, I adopted the lab team teaching model. And I wrote about this because it was really hard in a lab, as it is in a lecture class. When one instructor goes down, it's really hard to keep those classes going. So we have a team of teachers that often get the experience to overlap so they can do that class if somebody can't do it. And in addition to that, we have a wonderful, wonderful supportive staff that just are so help us, like I mentioned. If I ask them for 100 mils of this reagent, they will go do this. I have never felt supported. But because I'm supported, I'm willing to think out of the box and what can we do, because I don't have to worry about the constraints of that. When I'm supported, my thoughts are freer, and I can think about how can we deliver this material and content better. I think I can say that your bioeducation has prepared you well. And Apeksha, who is a Fulbright scholar, completed her MBA and is set to do on a different path in bio, says, I've been able to use the same analytical methods I picked up in lab to teach third graders English, put together board presentations, and even build healthier routines in my lab. And to put more weight into these things and these qualities that I've been talking about, I borrowed a slide from our Career and Professional Development Center. And they said there, what are the professional competencies that employers seek? And that is problem-solving skills, analytical, teamwork, communication skills, initiative, strong work ethic, technical skills, leadership, flexibility, and detail-oriented. I write well over 800 to 900 letters of recommendation a year. And I tell my students, who are usually shocked, that the first question, and I give about 50 phone recommendations for industry, and I tell them, you know what the number one question is asked of me? It's not what's your GPA. No one cares. If they're looking at you, they've already looked at your record. The first question that's asked of me is, how well will this person fit in my group? Are they disruptive? I'm not going to put anybody who's not going to get along with my well-oiled team. That's the first quality. And that's why I write so many letters of recommendations, because lab, we're in there 12 hours a week. We know you. I can really write those examples where I've seen you do that problem solving or designing that experiment. This is what we strive for. These qualities are what employers are looking for. This is what we highlight in our letters of recommendation. So what is your next adventure? Fit in last year's class. Typically, 53% are working, 29% are going on to continuing education, whether it be grad school, medical school, and we had a few people go to law school. 13% were other, and 5% are still seeking jobs. No matter what your journey is, we hope it's smooth sailing. Sometimes our journey gets a little rocky. Whether we are a faculty member here at CMU, honing in our craft and always trying to increase our problem solving, our analytical and reasoning skills, and you're a student building all of these qualities that we can write about you in a letter of recommendation. Remember, no matter what, this is why we're all here. This is why we all work at CMU, because all of these skills, this is why this was Bill Brown's vision. This was our problem-solving skills. This was Andrew Carnegie while he founded CMU. It's a problem-solving university. And to do that, we need all of these qualities. And this is what we strive for in every single class, and not just the lab class. In summary, CMU is very powerful teaching, interdisciplinary research, public service, and a persistent curiosity about all things. It's the place where passion and learning happens, and it's always been my privilege to see this happening in Pittsburgh and to come here and see the same passion and curiosity at our students here. But everyone, take a step back and realize we're all a community. We all work together, both our faculty, our students, and our staff, to produce well-trained, analytical, and problem-solving students. Thank you.
Thank you, Carrie. Thank you for this nice, inspirational talk. I would like to open the floor for questions from the audience. Maybe I can start. The biological science program runs labs in a different manner compared to many other biology programs in many other universities. So can you explain the difference and maybe tell us why do you think this difference benefits our students?
Okay, so I'll give you a perfect example. I was at the ABLE meeting, the American Biology Laboratory Educators meeting, and I met a laboratory director, an equivalent position at a very large university who at any one given time runs 20 sections of lab with 24 students in each section. And for gels, what they do is they come in with one gel apparatus, the grad student loads the gel, and then they hand everybody the gel image, and their lab is analyzing that gel. I want to work in a place where students pour their own gels, have to know what kind of gel they need to pour, and have that equipment and decide and prepare their samples and that's the benefit of our labs.
Great, thank you. Questions? Yes, Adviti.
Thank you, Carrie, for that talk. In the age of AI and cloud lab and all of that, where do you see lab education going in the future? I mean, Bill Brown had his vision to get up to here, but we're not living in that age where, technology is progressing and students have that support. So not really, avoiding using AI, but can we use AI to support, can we use technology to support our lab education better? So what is Carrie Doonan's vision for lab education in the future?
I'm not sure I have a well thought out answer. I've just started thinking about this in the summer. I'm going to some meetings about this. I do think there is a place for AI, initially as a supportive measure, but when the AI takes over the entire student's body of work, so there has to be some type of balance that the student can analyze, but it's not taken over. I'm not sure that's a great answer, but I think it's something that needs to be looked at.
I would like to piggyback on Adviti's question, since you mentioned the cloud lab. So CMU has the unique cloud lab, which you can run your experiments remotely, and everything runs in Pittsburgh while you are just typing codes on your computer, which is quite fascinating. I just wanted to get your thought on whether you think this can enhance or take away from students when it comes to their lab experience.
I think the cloud lab, and we do eventually have plans once what we're doing has been worked out. I think there's a few more things that have to be worked out before that. I think at a 100-level lab class, we need to make sure our students fundamentally understand the basics and what an experiment is. And then at the 300-level lab class, where we already know how PCR works in that, then we can work and do our computer programs and that to design and have the lab be a very important asset for us to use.
I see. Okay, maybe one last question. Yes, Andrew.
Hi, I interviewed Yasser a few weeks ago to talk about the faculty exchange last semester when Emily Drill came here and Yasser went there. And I was wondering if you could talk about the benefits that you see of doing that sort of thing.
I think that I'm a big supporter of that. In fact, I strongly encourage my lab teaching team to do that. I think it really helps. I'd first like to point out that, as an example, Emily and I are very, very different. I'm much more this, and Emily's much more this. That's why we're a great lab team. I will say, Emily, I have this crazy idea, let's do it. And she'll be like, okay, Carrie, we'll just do it. We'll see if it works. Sometimes it works, and sometimes it doesn't. But those different perspectives are so important that students get to learn. Emily's background is in neuroscience. I'm an old biochemist. But when a student comes to us in the 300 level labs and brings us a problem, and Emily can, and we're doing neurobiology of neurons, and I can explain the biochemistry part of it, and Emily can explain the neuro part of it, students get a better depth and perspective. And the most important things that students can learn from teaching in different places is that no one perfect way is right. Somebody might do it this way for this reason, and somebody might do it this way for this reason. But I think it is so important to foster that in both places.
Well, thank you very much. Let's give a round of applause to our speaker. I would like to call back Dean Mike Trick.
Well, thank you very much for that very interesting presentation. It is really good to know how unique we are in the labs and how amazing our biology program is. And so thank you very much. As a memento of your presentation, we do have a little gift for you, and so we can do that, and then we'll look over here, and they're going to take our picture. Good. Thank you again so much, Carrie.
Thank you.
Okay, thank you all for coming out today for the Brown Lecture. We have, I think, a light lunch available outside, and so you're welcome to join in the conversation. Thank you all for coming out.